particular expertise is showing the connection that I can tell between organisms like Chlamydia pneumoniae, endothelial dysfunction, and neurodegeneration. So with that, Dr. Balin, I'm very excited to see what you have to say and updating your research and the broad spectrum you can give us on infectious etiology of disease.
So this is who I am. And the pathogen hypothesis is something I've been working on for better part of about 25 years. So as we go forward, I just wanted to make the declaration that I have no conflicts of interest here, and that this work has been done in collaboration with a lot of people, as you see here on the screen. I have a number of funding sources that we've had over the years. Funding has been an issue, though, as a continuation of that, based on the fact that we've always been on the cutting edge and pushing the envelope. But all these collaborators...
In my opinion, Brian, Leslie Noren should have written you a check for $1 million.
Yes, but he didn't.
I know that.
That would have been nice, but I know Leslie, and yeah, I think he's doing good things, though.
Yes, bringing some attention to it. We'll
yeah, absolutely. So the learning objectives really for my presentation are listed here. And I'm going to go over some epidemiological issues, classification. The pathogen hypothesis is really the focus. Neuroinflammation and the crossover with the amyloid cascade hypothesis, and then with the Chlamydia pneumoniae story, which I've been working on for and first published in 1998. So here we have the classification. Sporadic Alzheimer's disease is really what we're talking about for the most part here, where it's classically defined as something over the age of 65, late onset. Familial disease does occur in a small percentage of individuals that get Alzheimer's disease, and this is really due to genetic mutations. It can show up in the 30s, 40s, and 50s. There are a number of different communities across the globe that actually have demonstrated they have familial Alzheimer's due to a number of different issues, which I'll go over shortly. The risk factors. The major risk factor really is age, is the number one. You do have genetic risk factors as well, especially the apolipoprotein E apolipoprotein. So a apolipoprotein that actually moves cholesterol in and out of cells, and especially the epsilon 4. There are 5 isoforms of the ApoE apolipoprotein, but the 2, 3, and 4 isoforms are the most commonly found in the human population. And the 4 gives you, epsilon 4 gives you that increased risk. And 3 is also quite obviously people with an epsilon 3 genotype and phenotype actually do go on to get Alzheimer's disease. The epsilon 2 seems to be somewhat protective and decreases the movement of those chylomicrons and cholesterol and lipids into cells and out of cells. So it keeps it more soluble and into the bloodstream versus going into tissues. The other risk factors I have listed here: atherosclerosis, diabetes, head injury, toxins, infection. A lot of these are being studied now. Head injury is a big issue, obviously, with the chronic traumatic encephalopathies that are observed and now increasing risk to go on to a degenerative disease later in life. And infection's been our major focus. I often get this question: what is the difference between just a typical age-related change versus Alzheimer's disease as far as the symptomatology goes? And you can see on this slide that signs of Alzheimer's and dementia are pretty drastic, that are consistent as well. When someone is diagnosed with this disease, they typically will start off with these types of problems, and then it progresses over periods of time. With the typical age-related changes, these are very infrequent events, such as missing a monthly payment of a bill, forgetting about it, or remembering something later. Sometimes forgetting a word to use which we all suffer with as we age. You're looking for word searching at times, and that can be an issue. So these are now things that are correctable versus Alzheimer's disease. So here's Dr. Alois Alzheimer, the psychiatrist with the microscope. What you're seeing here, this micrograph is actually a silver stained image of August Dieter's brain, the individual that he first followed, came to him severely demented, followed her till death, did the autopsy of the brain and neuropathology using silver staining, and found these plaques and tangles actually showing up in her brain. Years later, by the way, this happened in 1906, 1907. In the mid-1990s, tissue was still available from this particular individual to be analyzed, and they found that she actually had a genetic issue with presenilin genes, the presenilin 1 gene, and it was a genetic issue that this individual had actually, that Alois Alzheimer first identified. And at the time, he had another individual, another neuropathologist, Oscar Fischer, who was doing the same type of work. Alzheimer happened to publish his work about 6 months earlier than Fischer's, and that's we end up with Alzheimer's disease versus Fischer's disease, which is interesting. Emil Kraepelin was the institute director where Alzheimer worked, and due to political and economic reasons, he actually now named it Alzheimer's disease for Alzheimer, in case you were wondering how it became known as Alzheimer's. The brain atrophy in advanced Alzheimer's disease. So on the left-hand side of the slide, you see the normal situation here. The brain on the lateral view at the top looks very compact. The gyri look fairly normal. The sulci, the spaces between the gyri, are very compacted. Then in the coronal section down below, you see all of the major features. If you actually look down below in the temporal gyrus, or a temporal cortex, you can see the hippocampal formation nicely on each side. If you go over to the Alzheimer's brain at the top, you see now it looks like the gyri now are shrunken and the sulci are widened. And that's because the mass of the cells being lost. So you're actually getting cellular loss here. And the cellular loss is dramatic. A typical brain weighs about 1,200 to 1,250 grams. With Alzheimer's disease, the weight of the brain can drop down to 1,000 or 900 grams. You're losing a significant amount of material. And then with the coronal section, you see down in here with the temporal lobes, the hippocampus, where it should be, is gone. And the ventricle system is enlarged. That's what you see here with the eye-like structures here in the mouth below. So those are the lateral ventricles and the third ventricle. The Alzheimer's pathology. Neurofibrillary tangles and neuritic senile plaques are the key pathologies here. I do need to tell you, though, that you can find other pathology in Alzheimer's disease. You can find alpha-synuclein inclusions, a different type of protein. You can find TDP43 inclusions as well in nuclei, in some cases of Alzheimer's disease. So you see some other pathology that shows up. But characteristically of Alzheimer's, you find these tangles that are made up of this hyperphosphorylated tau protein, which actually is a microtubule-associated protein. In fact, early in the 1960s, early 70s, Alzheimer's disease was thought to be due to a microtubule deficiency within the cells, leading to abnormal communication. Well, it is a problem in the cells, but it's because of this hyperphosphorylated tau that starts to accumulate into these paired helical filaments, forming then into these large tangles. And over here, you can see a neuron that has a tangle in it, and then over here, neurites down below that are dystrophic within this plaque area. And then the most characteristic thing that a lot of people point out and have been studied widely in this disease, both of these entities have been, are these amyloid plaques made up of beta amyloid peptides. And this is an abnormal accumulation of broken down protein, the amyloid precursor protein, into these very small pieces that start to aggregate together. And this involves different types of proteins and gene expressions. And we'll go over this next with this particular slide. In this slide, what you see is a non-amyloidogenic type of process versus a pro-amyloidogenic process. And if you look at the schematic, starting at the left, you can see that you have three major secretases. It's alpha, beta, and gamma secretases. The alpha secretase actually cleaves this amyloid precursor protein in a way that doesn't allow for any accumulation of proteins or peptides to occur. But if you get a beta secretase and gamma secretase secretion and cleavage events, it does allow for these peptides to start to be produced. And by the way, they're first actually showing up inside of a cell, and then they're expelled outside of the cell and aggregate outside of the cells in the brain tissue proper, leading to their accumulation into these plaques. And those are the characteristic of the amyloid plaques. And we just have now here the different enzymes that are involved. In familial disease, the beta secretase is functional, and the gamma secretase is abnormally functional. So the presenilin genes actually become mutated, leading to their increased activity in familial disease. In sporadic or late onset disease, we actually don't know why you're getting increased activity. We believe, though, that you can trigger that increased activity through infection. So this is where, and through neuroinflammation, and this is where the story starts to come together, we think. So what initiates this plaquing and deposition in sporadic disease? Two hypotheses we believe are key: amyloid cascade hypothesis and the neuroinflammatory hypothesis, or neuroinflammation. The merging of the hypotheses here, we think, is infection, because infection, we believe, is actually a major trigger for the neuroinflammation and chronic inflammation, which we talked about a little bit earlier, and persistent inflammation. So something that is happening here in a chronic way. And we think infection is the driving force for that initially. Once that's starting to occur, though, you will get amyloid over here deposited. And so amyloid deposited in an abnormal way is also becoming a driving force maintaining your chronic inflammation. I've often described this as a different type of entity. And all of you on the call today may know what a granuloma is, like in tuberculosis, where you have now an accumulation of cells that are interacting with one another, leading to massive damage and what we call the granuloma, often in TB, in the lung tissue. Well, amyloid plaques remind me of granulomas, where you now have an accumulation of cells and debris and amyloid accumulating, leading now to a constant nidus for inflammation. But what is really stimulating that initial inflammation? And that's where we go backwards to the pathogen hypothesis. And this is what that actually means in our minds. These microbes, pathogenic microbes, depending on their ability to enter the brain or trigger inflammatory responses, can lead to the activation of the pathways that lead to the generation of amyloid and eventually the neurofibrillary tangles that occur, demonstrating that neurons are damaged and irreversibly damaged over periods of time. That gives you then the significant progression of this disease process. The proposed infectious agents. So here's what we're talking about. So the Chlamydia pneumoniae story that we first published on in 1998. So we were the first to put that together. It took about 2.5 years to gather all the data and evidence for this. And then over the years, we published more.
Shower. Let's get in the shower.
The other organisms that obviously have been included in this, Borrelia species, especially Burgdorferi with Lyme disease, and Judith McCloskey has been a big proponent of that. Helicobacter pylori with different individuals that suggest that this organism also is on board in individuals with the disease and can potentially lead to inflammatory issues in this disease, potentially leading to the degeneration that we would see. Another one that hit the scenes more actually recently was Porphyromonas gingivalis. These people actually did a clinical trial trying to now inhibit the gingipains that are made by this organism. And the clinical trial really hasn't been successful. But they think that this organism can be an initiator or a contributor to Alzheimer's disease. Other oral organisms, treponemes, for instance, gut microbiota, these have been included in the discussion as well. And in some evidence has been produced that they could be involved too. Candida species, Toxoplasma gondii as the parasite that commonly will show up in individuals and pretty frequently in a number of individuals. Herpes simplex virus 1 is probably the most noted virus that has been associated with Alzheimer's. This probably is a player in the process. Human herpes virus 6 and 7, probably less so, but could be. And then we go to COVID-19 and SARS-CoV-2. Well, just recently there was an article out suggesting and showing that there's an increased risk in individuals over the age of 65 that have contracted COVID-19 or this virus. There's a 50 to 80% increased risk in those individuals that have contracted that virus of now getting Alzheimer's disease or being diagnosed with Alzheimer's disease. So that's pretty significant. And it sort of now is bringing the attention back to many of these earlier studies that many of us have been focused on. Well, let's go to the Chlamydia pneumoniae story a moment here. It's a ubiquitous respiratory pathogen. It's implicated in numerous diseases, typically community-acquired pneumonias, but will show up in atherosclerosis with systemic infections as well. It's an obligate intracellular bacterium, meaning it needs to grow inside of our cells. It cannot grow outside because it requires ATP that it siphons from the mitochondria inside of our cells. And also it gets other nutrients such as tryptophan and carbohydrates and lipids from our cells. So it's an intracellular bacterium. Serological evidence would suggest that about 75% of elderly individuals actually have been exposed to this organism. You can do serology where you're finding IgG and IgA evidence for that. This is just now a summary of our first publication that came out in 1998. And you could see in 17 of 19 brains, 80 brains, we had PCR positivity for the organism, which you see over here with electron microscopy, by the way. The arrowheads are pointing out the elementary bodies, which are in the infectious form of the organism. And those are the arrows. And then the arrowheads are pointing out the reticulate bodies, which are the actively growing forms of the organism. This was an electron micrograph taken from a sample of an Alzheimer brain that we evaluated. And then for the control brains, 18 of 19 were PCR negative. So neuropathology was in the areas where we found the infectious agent. And this is a light micrograph, an immuno-labeled micrograph of a neuron down below here. That brown area there is labeling for the organism inside of a neuron. One of the things that when we were getting ready to publish this work, we were asked by some editors of journals to actually see if you could actually grow out the organism from frozen tissue. And the reason being is that we felt that in frozen tissue, this organism in the elementary body stage, where it's more of like that infectious form that's more like a spore, that it could still be alive. because this tissue was frozen, and that if you put it in culture, potentially you could grow it out, which is exactly what we did from a number of different brains, Alzheimer brains. And so by light microscopy A, B, and C, you can see these different dark material in there is evidence of the organism. By EM down in D and E, you can actually see the organism within monocytes from our cultured situation. Many evaluations of our human brains have taken place. What we have found in summary are this, is that glia, both microglia and astroglia can be infected, neurons can be infected, and also endothelial cells. So this organism, which is interesting, can be, once it's inhaled, can get into our brain in a couple of different ways, and also affect just about every cell type that it's exposed to. So the brain areas affected, we focused in on entorhinal cortex and the hippocampus, and sure enough, we found a lot of evidence for those areas. The entorhinal cortex is where your olfactory pathways come into the brain. The olfactory tract is one of the areas that is now directly connected, and synapses are present from the olfactory bulb in the entorhinal cortex, where you're getting a processing of sense of smell. The other areas are the amygdala and the orbital frontal cortex. All of those areas show evidence for infection in our analysis. Also, all of those areas are some of the earliest areas affected in Alzheimer's disease when you look at very early onset of Alzheimer's disease. So the entry, we believe, is through nasal neuroepithelial cells in our upper parts of our noses. So that olfactory system is involved, and also through the blood-brain barrier. We think we actually have a two-hit hypothesis here where you can get infection from both sites through olfaction, uptake through sense of smell, and also through the blood, where the organism would be carried by white blood cells around our circulation. So here's a study that we did where chlamydia pneumonia was detected in the blood from AD and MCI patients. So early Alzheimer's and mild cognitively impaired patients. And what this is demonstrating is that you have three forms of the organism: the elementary body form, the intermediate body form, and reticulate body form present in these monocytes that were now carrying the organism in the bloodstream from these individuals. The greatest form of this organism was the elementary body form, which is in green in these graphs, and then the reticulate body forms in red, and the lesser form here, but the most probably problematic form is in blue here. That's that intermediate body form, which we believe, and others do, that it could be the persistent form of the organism that actually now can stay around for a long period of time in our cells. So routes of infection, as you see through this diagram, the green dots going into the brain and then going into the lungs. So the respiratory infection leading to systemic uptake in the blood, and also through the olfactory pathway. What's interesting is sniffing out Alzheimer's disease. Over 20 years ago, Dev Devanand showed that that there's an inability to recognize certain smells in individuals that have Alzheimer's disease, and they're unaware that they have this impaired odor perception. This smacks currently of what we find in the COVID infections, where the sense of smell is being impacted in a large percentage of the population. That's why we think also olfaction in COVID is a very big issue and may be the key to why we're increasing risk in Alzheimer's disease in older individuals. So we've gone about looking at the pathways for proving a pathogen involvement in Alzheimer's disease. And this is just a flow diagram, basically, showing you different techniques that are used to try to demonstrate that this infection not only is present in the disease, but actually could be causative for the disease. And this is where we're focused upon. And causation because of the generation of amyloid, accumulations of inflammation in the brain tissues, of a persistent insult leading to a chronic type of neuroinflammation in brain tissues. So if this organism gets into our brains, this could be a really source of something that is continually leading to an event or events that are continually now propagating or leading to the propagation of amyloid and eventually neurofibrillary tangles. So our mouse studies, just in summary here, we actually took normal BALB-C mice. These are not transgenics. These are normal animals, young animals, actually about 3 or 4 months of age, and just inoculated them with Chlamydia pneumoniae in their nasal passages. It's picked up in the olfactory neuroepithelium, the nasal epithelium. It also gets into the olfactory bulbs, as you see in the bottom micrographs here. These are electron micrographs showing you these dense bodies, which are these organisms of chlamydia present in the olfactory bulbs. So it's a proof of principle and concept that infection through olfaction can actually get into our olfactory regions of the brain. If we now go further and actually look at the brain tissues of these individuals, we'll see that you can actually have amyloid plaques. These dark entities here in the brain tissues of these individuals, of these mice, are actually amyloid A beta 1 to 42 plaques. So they label with antibodies to A beta 1 to 42. So we're generating the pathology that now we have found in humans in the mouse brains. You can also then go on and recover organism from brain and olfactory bulbs. It's easier to recover in different aged animals from the olfactory bulbs than brain, but you can still recover it. And we found that if you use a AD brain strain isolated from Alzheimer's disease brains and respiratory strains of the organism isolated from lungs of individuals that had the infection in their lungs, that both of these strains can actually lead to the infection. The high dose infection actually gives you a more prominent uptake and recognition of the organism in brain tissue. The length of infection, the longer the infection goes on, you'll also find increased organism present in brain tissue. And the age of the animal. Young animals seem to be somewhat resistant to being infected versus old animals. So we believe, and over here you see in the graphs, these are old animals. These are higher titers in the older animals versus younger animals, suggesting to us that the age actually does have an influence. So what we did here was we took 6-month animals and 12 and 20-month animals and infected them, and realized our 20-month animals actually are very subject to being infected readily. If you think about us in the human population, older individuals, why is age a #1 risk factor? Well, we think partly also because we're now more subject to being infected and getting these infected agents in different parts of our bodies as we're aging and when we're older. The blood-brain barrier. So going back to the blood-brain barrier moment, so we realized that in human brains, you could pick up the organism in human brain endothelial cells. So it was present there, but it was also present in monocytes in humans. So we did a modeling here in vitro, where you took a blood-brain barrier model and evaluated transmigration of white blood cells through an in vitro blood-brain barrier. And what we found is if you infect your human brain microvascular endothelial cells, that transiently the junctional proteins actually are now diminished. They actually now decrease their expression when the endothelial cells are infected. And then if you have infected monocytes along with those infected endothelial cells, the monocytes bind on to the endothelial cells and migrate through this barrier. What this tells us, if we translate it to the human condition and human brain, it tells us that the infected white blood cells that can now be picked up or pick up infection from our lungs, when they circulate around, they can also now bind onto the endothelial cells, potentially, of our brains, and now be carried into the brain in a white blood cell. That smacks of actually how HIV gets into human brains when you have HAND or the HIV-associated neurodegenerative process, because it's using white blood cells as well to get into the brain that way. So this goes to that two-hit idea that we have of olfaction and blood involvement through the white blood cells coming through the blood-brain barrier. Other studies that we've done here is look at cellular mechanisms. So what's very interesting here is autophagy gets initiated when you have infection of Chlamydia pneumoniae in cells. Also, that once that's initiated, by the way, we think that is involved with the generation of amyloid, the now uptake of the transmembrane protein of amyloid. As membrane is now being endocytosed into a cell, it now is being degraded through the lysosomal process, and now is leading to the abnormal cleavage events of your amyloid precursor protein into those amyloid peptides. And then once it happens intracellular, those vacuoles are now pushed outside of the cell, leading to the deposition of your amyloid in the extracellular space. The other interesting thing that we realized and actually studied and published on is how apoptosis is inhibited. So if you have infection of cells, it will actually shut down the apoptotic process inside of the cells. That's how chlamydia stays more of a persistent chronic type of infection in our cells, and we think in our bodies over time. The other things that happen, calcium dysregulation. Chlamydia pneumoniae is a sink for calcium and also for iron. And you'll find that when you have these infections on board, that iron accumulates inside the cell as well as this calcium. And calcium then, when that gets dysregulated, you're leading now into cascades, leading to phosphorylation events, leading to breakdown of membrane through activation of phospholipids, etc. So you really have a problem here with calcium being influenced by infection. The other thing we've looked at is inflammasome activation. Inflammasomes are inflammatory expressions inside of cells where you're generating IL-1 beta for the most part, IL-18 and IL-33. And this is all through the activation of your transcription factors, the NF-kappa B transcription factor, leading to the production of these cytokines. And this is, we think, how the pro-inflammatory response is generated when you have this infection on board, leading to that early inflammation that's occurring actually before amyloid is generated. And cellular mechanisms in monocytes, also inflammation is occurring. So when those monocytes are infected, we published on this study that you can actually find that IL-1 beta, IL-6 and IL-8 are produced. Again, the pro-inflammatory cytokines that are leading to now, in this case, it would be systemic inflammatory responses that could also influence how the blood-brain barrier would be functioning. Astrocytes. What we realized is that, and a number of people years ago realized this as well. So we were not the first to do this, but we're the first to infect them and to find the result by infection with the Chlamydia pneumoniae story. But astrocytes can also generate amyloid. And while they are supplying nutrients and neurotransmitter factors like glutamate for neurons and to keep them healthy, and also regulating blood flow, if they're infected, they can actually be a source for a dysregulation of glutamate and also dysregulation of amyloid and production of amyloid, which is real interesting. So in culture, we did a study where you can actually infect them And on the left-hand side of the slide, you see cells that were not infected. On the right-hand side, they are infected, and you can see these yellow, greenish type of structures inside the cell. Those are now the vacuoles containing the Chlamydia pneumoniae organism. But you can also see here, it's been labeled for amyloid. All that red inside these cells is amyloid that's actually being generated when you have these infections on board. And you can compare the right-hand side of the cell or the slide to the left-hand side, and you see you have much more red inside of these chlamydial infected cells. And if you do the measurements here for beta secretase and gamma secretase, protein expression, you'll see that both beta secretase and gamma secretase in the greens on both sides here are upregulated through 72 hours post-infection. And that tells us that the infection actually can lead to increased expression of those genes that are now coding for the base and presenilin-1 proteins. As the proteins get upregulated, they're now increasing their functionality. This is why we believe amyloid is being generated in the infected cells. Linkage between the Chlamydia pneumoniae infection and other risk factors. There is a linkage we've discovered between ApoE epsilon 4 and C. pneumoniae. In a study we did in culture, if you block uptake of ApoE4 in the presence of C. pneumoniae, so if you block the LDL receptor protein where ApoE4 binds in the presence of Chlamydia pneumoniae, you prevent infection. So we actually believe that the ApoE story here is more complex than what we've been led to believe up to now by other reports, that it actually is involved also with infection. By the way, Ruth Itzhaki found this to be the case also with the herpes simplex virus 1 infection. There is a linkage between this particular apolipoprotein and the epsilon 4 isoform with these organisms. The other types of linkages, atherosclerosis is a biggie here that's been found with Chlamydia pneumoniae. A lot of articles, hundreds, it actually goes into the thousands with atherosclerosis. Diabetes. There's a pathogen burden in diabetes, shows a strong association with insulin resistance. McLossie and McGeer actually published this in 2016 and found that they found Chlamydia pneumoniae on board in this regard as well. And in Alzheimer's disease, insulin resistance is a huge issue because it does occur, and it's not well understood why it's occurring. And we think because infection is on board, that it's actually leading to the insulin resistance issue. And of course, neurotrauma as a risk factor, anytime you have blood-brain barrier disruption, any infectious agent that would be in blood or that you're now being exposed to, it gets into the bloodstream, could now invade the brain. So now I have a little summary here, comparing Alzheimer's disease and Chlamydia pneumoniae infection on different sides here. And what you see with Alzheimer's disease, I have all of these dysfunctions noted. Now, this could be just coincidental that all of these dysfunctions also correlate with what a Chlamydia pneumoniae infection actually does and can do. But we think it's more than coincidental. We think that this is actually now tying the knot between the two of why there is an association of this type of infection with this disease and why it actually can be a trigger for the entire disease process. And this is that proposed mechanistic model. If you go from the left, starting with infection and invasion, we have a number of different ways that this is occurring through the neuroepithelial cells, the olfactory neuroepithelial cells, and through infection of white blood cells, going through blood-brain barrier, et cetera. So a two-hit hypothesis. Cellular dysfunctions that I've mentioned already, many of those happening. And then eventually this abnormal amyloid, neuroinflammation, I don't have here the neurofibrillary tangles extended from this, but they would come on a little bit later. But this would all lead to massive dysfunction in the brain over time. And then going back to that last slide, which I had up at first, with Alberts and Georgi, discovery consists of seeing what everybody has seen and thinking what nobody has thought. And he actually won 2 Nobel Prizes, I believe, for different findings throughout time. I think one of them was on a vitamin itself. So I'll stop there and entertain any questions.
Dr. Balin, you can get off the screen share so we can see you. And I just, since this subject is so near and dear to me, there's a bunch of great questions out there, but I'm going to ask you a few first.
Okay.
First to our comments. First of all, Dr. Tremp and I, in our book, The End of Alzheimer's, written before Bredesen, mine's more of a textbook, but we posited that the hyperphosphorylated tau is actually protective because it's found in reverses in hibernating animals. Now, that's the only shred of evidence I have, but a commonality between hibernation and Alzheimer's is hypoxia potentially. And so it's there as sort of a protective shield to the hypoxic brain. And any thoughts on that?
That's interesting. I haven't really considered it that way before, that it would be protective. And the reason being is that when you start to see these tangles accumulating in these cells, it's an association also with synaptic dysfunction. So, you know, if it's trying to preserve a neuron, but it's also leading to that dysfunction, that connectivity dysfunction, I don't know how it would be protective in that sense.
Yeah, I don't either, but usually these phosphorylated compounds have some energy mechanism or maintenance in them. But I saw the association. I wrote about it like 6 years ago and have left it on the shelf since then, because what the heck can we do about it? But the only evidence I have now is that Tau RX came out, and they came out when I was writing this, and I predicted they would fail, and they have failed. So just down-regulating the hyperphosphorylation process doesn't necessarily provide any benefit. And then Tansey talked about the beta-142 as being an antimicrobial peptide.
Right.
I haven't seen any subsequent follow-up on that to any large extent. Any comment on that?
Yeah, so I actually believe that it can act that way, especially with the extracellular bacteria and maybe some fungi as well. We actually did a small study after talking with Rudy Tansey and Rob Moore, who is deceased now, but he was the one driving that process and progress in that field. And found that when you interact amyloid with the chlamydia organism, it doesn't do anything. In fact, chlamydia actually is generating the amyloid by infecting cells, is generating the amyloid. And one of the ideas I had about that was, indirectly, that may be a response to an infection of like trying to keep other infections from bothering that cell. You know how infectious agents can make antimicrobial agents so that they're not impeded upon.
Well, cholesterol like fire truck shows up at the scene of the atherosclerotic plaque, but it still doesn't save your life. You still have a heart attack.
Right, exactly. So it's sort of like this material, it can have a function, but as it accumulates as well, it goes from potentially good to bad over periods of time. And I think it is a nidus of bad over time, but initially, potentially it is an anti-infective molecule, or can act that way. Just like LL37, also expressed by microglial cells. It's very antimicrobial in nature as well. And they can act in a similar fashion. They're different though, but.
A couple more comments and I'll try to get some of the questions answered, but so Dr. Tremp told me that in the 1990s, he stopped doing IgA and IgM because IgG told the chronic story. And subsequently, I read Ewald's book with, real like a fine-tooth comb, try to figure out what he was saying. And he seemed to be saying the same thing. And a number of other people have indicated that IgG is an indication, not a historic. And then Dr. Carter and I were blessed with a corporation that allowed us to work on 100 people. 41 of them we positive were infected, and we ran detailed infectious panels on them. And sure enough, they would have, some would have IgG, some would have IgG, IgM, some would have bacteria, some would have, they often have a comorbid viral, like Isaki talks about. But when we treated them, they pretty much all got better. We used what we call the TREMP protocol, which is like the Weldon protocol. Biaxin, which downregulates, it's antibiotic, but downregulates nuclear factor kappa beta, and then minocycline, which is a lipophilic tetracycline. And by the way, Mark Nelson is speaking tomorrow night. He formed a company, Powertech Pharmaceutical, with Wally Gilbert, Nobel Prize winner. He's probably the top tetracycline scientist in the world. But
yes, I know Mark.
Okay, good. Right, I introduced him to the intracell group.
Okay, that's how I met him. I actually
right now it all makes sense. But when Dr. Carter and I and Dr. Trent Austin treated this group of 41, those that had IgM and IgG, they both came down. Those that only had IgG, the IgG came down. So that's a long prelude into, in these different forms that Stratton talked about, is it possible that IgG can be up and that it's not a historic infection, it's sort of a stealth or a chronic phase or spore form of the infection, but IgG is telling a story that the infection is there?
I think so. I would agree with that. I think that's the case, because we've seen more IgG than IgA expression also. Exactly. Every now and then we'll see an IgM, but it's usually with an acute infection. So the IgG is more maintained, I think.
I think the chronic 99.99% plus 0.01%. So 100% of infectious disease doctors will say IgG is historic and irrelevant. But see, what Dr. Tremp told me as an eye doctor, that he read the literature like no one, and as a practicing clinician at Scapins, both in research and in the clinical delivery, that if you have a toxo scar and you do a blood test and it's only IgG positive, IgM negative, and you extract that scar and put it in culture, it grows.
Interesting.
Yeah, I can't find that study. He passed away on me like Stratton and Weldon did, and his widow sent me like boxes and boxes and boxes of all his stuff. And I know that study's in there somewhere. He didn't do it. It's out there in the literature. But anyway, that's that. And then, I read the guy who introduced me, this crazy doctor that introduced me to Dr. Tremp, showed me this paper in 2006 that, and you kind of mentioned it, that these organisms, like it can change apoptosis, they actually modulate the genome of the cell. And I'm sure you've seen that. And if not, I'll send you the papers I have on it. Infecticogenomics.
Yeah, I believe that. And I think that's probably what's happening as well. Also, they're influencing, the expression of certain proteins important for mitochondrial function. What we're finding with the chlamydia story, and probably other intracellular bacteria too, that the mitochondria are subject to damage because of the stress on them, because of how the organism actually is now sort of taking over the cell and really parasitizing in that effect.
Is there an uptick in superoxide dismutase or anything like that in these cells that are infected? And is there any way to measure that?
I think there's a way of measuring it, but we really haven't measured that with the superoxide dismutase.
Melatonin, glutathione, cellular respiration, and oxidative stress. And these organisms clearly put a burden on that whole process. I think it's like throwing the turbo on. Anyway.
Oh, yeah.
Just something I've been thinking about. And then I kind of, two last things, and I'm going to answer Dr. Stark at Harvard's comment question too, which I'd love to have you chime in on.
Okay.
Is one on, when I read Ewald's book initially, he said something about the number one way to prevent spread of infectious disease is through hygiene. I said, no, it isn't. But then I realized he's 100% right. And you spoke to that very clearly. nasal and lung infiltration.
Yes.
And like, so I mapped out the curve of tuberculosis versus time for like 1900 to say 2000. And it's coming down very rapidly, 1910, 1920. Then when antibiotics first came into play, like in 1950, and there wasn't even a blip to the downswing. And then the vaccine, like in the 1950s, not even a blip. So the entire control of tuberculosis was completely hygiene-based.
Yeah. Yeah.
So when people get all scared about these organisms, it's like, clean your nasal passages, clean your mouth, make sure your gut has strong acid, and do some nebulizing in the lungs once in a while. That might be the best way to handle these things.
It might be, actually. Yeah, we focused in on the olfactory pathways early on.
Sure.
Because of the respiratory nature of the chlamydia organism. And also that's why I think Ruth and Saki's work too is so important, because from the trigeminal ganglion, there's a pathway up into olfaction as well, where herpes, typically sits in the trigeminal ganglion. Why it would get through olfaction into the brain. Because it dawned on me that with herpes simplex encephalitis, you will specifically have if it goes to that level, destroy the temporal lobe. And the question was, why always the temporal lobe? Also, it goes to the issue of it. I don't truly believe the blood-brain barrier is the first part of the problem. I think it's a secondary part of the problem. I truly believe olfaction is the key issue here, and not just with Alzheimer's disease. Parkinson's as well, probably schizophrenia as well, and maybe some of the other neurodegenerative processes like the frontotemporal dementias. And it's because you've got the amygdala involved, you've got the entorhinal cortex and hippocampus involved, you have the orbital frontal cortex involved, all of the key areas here leading to many of these diseases that we see in the human population.
And that's why the oral pathogens, if you have a root canal up here.
Yes, that's correct.
And it pushes into the sinuses. We've had many cases of that. So it may not be entering through the CNS. It might be entering through the sinus.
Yeah, it's possible.
The other thing is, so Dr. Stark is one of our followers from Harvard, and she asked about mycoplasma, but I like to use the example of asbestos. It's completely inert, yet it causes cancer. And how does it do it? Because it's shaped in such a way the immune system really can't get a finger hold into it and remove it. It's just creating low-grade inflammation. So in my opinion, anything that can find what Charles Mayo defined as sort of like a localized or focal infection that the immune system cannot really get to is going to cause some type of inflammatory affliction. But the specific question she asked was, do you see any data between mycoplasma infection and Alzheimer's?
In the literature, I believe there's some data on that. I don't believe it's as strong as other infectious agents, but there has been in the past some individuals noting that mycoplasma could play a role there. Obviously, the mycoplasma is a strange organism because it has no cell wall. But it's respiratory. Could it go through that way? Potentially. I would think it'd be a more difficult process to set up a longer-term infection in the brain that way. But I'm sure it could probably lead to probably a meningitis type of reaction.
Yeah. You might be happy to know, and I don't run on chlamydia, pneumonia, IgG, IgM, and everybody, but almost everyone I run a chlamydial antibodies IgG panel on. And I'd say about 10% come up positive on that. And my population is relatively healthy and relatively well-informed about health. They came to me through summits or whatever, because I don't advertise outside of that. So that's that. And I throw mycoplasma in there, not because I think it's so virulent. I completely agree with you that, when I read Ewald's book, there's sort of a, transmittability versus pathogenicity, and mycoplasma is arguably much less virulent. The only two instances of a real pathology, and this is out of hundreds of people, when their mycoplasma is like way above normal, 1,500, 2,500, I've seen pulmonary embolisms. And I don't know if it's mycoplasma, but that was the coincidence. But I consider mycoplasma relatively non-virulent, and I run it anyway for the point of having a conversation about so the person sees it, they're positive many times, much higher frequency than chlamydial infections. And so it's an opportunity to start an educational process with people. That's why I run mycoplasma.
Yeah, and that's a good point. The other thing with mycoplasma is that it produces cold hemogglutinins, and that's probably leading to the clotting effect that you're probably seeing with really high titers of that, because you'll get erythema nodosum also and erythema multiforme in the extremities, Stevens-Johnson syndrome, etc., with that organism at times. So I think it's probably because of high titers of that leading to that aggregability.
Yeah, I didn't dig down into it to find that. I have to look that up. And Dr. Stark says, what do we do other than antibiotics? Look, I'll just give my own personal story, Dr. Balin. I had atrial fibrillation driven pretty much, I presume, by Lyme disease. Mm-hmm. And looking at Weldon's protocol and Dr. Tremp's protocol, because I was persistent for so long, I didn't have health insurance and I didn't want to go for a procedure, but I let it go too long. Typical, right? But I treated myself with high-dose cod liver oil, minocycline, and roxithromycin, which isn't available in the U.S., but you can get it here, for 18 months, never missed a dose, never took a probiotic, never had gut dysbiosis. Then I got my ablation, and 12 years later, I'm sinus. And a lot of people go back into AFib because they didn't treat the underlying. So anyway,
interesting.
Stratton, Weldon, Dr. Tremp, we all have antibiotic protocols. Do you have anybody you think has a decent treatment protocol that's not antibiotic for an organism that is actually causing harm? For example, chlamydial, it's actually in the, causing harm. So it's been there a while and is really entrenched in your physiology.
Yeah, we've thought about this a lot. I don't have a protocol that's not antibiotic at this point in time.
Right.
But we've thought about potentially developing or talking to people about developing a phage approach, a killing phage approach. It's very specific for chlamydia. And so that would be an approach to take, I think. As far as another approach maybe is, we have found that when the organism is growing, actively growing, it's more susceptible to antibiotics.
Right.
So it's sort of counterintuitive here, but one of the things that makes it actively grow is feeding it tryptophan. It loves tryptophan. In fact, one of the responses of our bodies to the presence of the organism is to actually increase IDO activity, indolamine 2,3-dioxygenase, to break down tryptophan. Okay, and interferon gamma can also actually activate that process. And to break down tryptophan, it starved the organism. But in some ways, that may actually lead to its persistence in the body, because you're now driving it more into the elementary body form, which is very, it's not necessarily dormant, but it's very inactive. And
right.
that probably is close to the persistent form. So the other approach would be, what if you feed it tryptophan and get it to grow and use antibiotics at the same time to actually now eradicate all these growing forms?
See,
it's an idea.
Yeah, Dr. Stark, the trouble is with these different forms of the organism. I had this conversation with Dr. Kilman McCulley, and he's written more medical hypothesis papers than anybody I can imagine. He's 88 years old. It's hard to catch up with him. He's written so many. But I said, when they can, it's probably like someone can, hold their breath for a long period of time. They eventually have to come up. So our theory on this, and I don't do deep research like Dr. Balin, but the theory is that you need to keep a pharmacologically relevant anti-infective dose for these circumstances that you can't predict, when it may become vulnerable as it goes out of these different phases. And the other issue is, as my dog gets on the chair back there, The other thing is what we find, the reason why minocycline and clarithromycin, and I don't think it was a guess by Dr. Tremp, work, is because when the microglial get activated, sometimes even if you've gotten rid of the source, they can stay activated, just like in a concussion. Mm-hmm. So you have to have an anti-inflammatory component. So like, for example, hyperbaric oxygen or ozone, if you could do it every day for a year, wouldn't necessarily relieve the symptoms of a neurodegenerative process because the brain may still stay in this peaked inflammatory phase.
Yeah, you know, you make a good point there too. That's why we think that with the amyloid that gets generated and deposits, it's going to maintain a foci of inflammation. And so those microglial cells may still be responding to now this material that they're trying to get rid of.
Exactly. And
they can't. So even though you've possibly gotten rid of the infection or limited that, you now have the other ingredient. So what that tells us is that we need to do something very early on in the disease process.
Really?
If you have mild cognitive impairment, maybe earliest diagnosis of Alzheimer's, that's when you have the window of opportunity. If you miss that, the disease is taking over and you can't really correct it. It's like you said earlier with atherosclerosis. Once you have the lipids deposited there, it's almost too late.
Yeah, that's why my dad had glaucoma first, and then he had Alzheimer's, and it doesn't always happen, but the eye seems to be a canary for Alzheimer's. That's why I have everybody running an OCT. So we can see if there's any nerve fiber layer thinning, because the OCT is much more precise than an MRI. You know, the resolution is superior. The MRI doesn't start seeing atrophy until well along, but the OCT will see atrophy down to the micron level the second there's any kind of atrophy. So I tell people, look, and that's how Dr. Carter and I met, because he's had a long-standing battle with glaucoma, is that there are papers that say glaucoma is Alzheimer's of the eye and vice versa. Not always focal infection, but at least get your eyes tested. It's an easy screening tool. And that's how Dr. Tremp became what he became really in this neurodegenerative field. For the people that at least knew him, is, you know, he was reversing, and I have a paper I should have published 5 years ago, but I'm still sitting on it. He would reverse retinal nerve fiber layer thinning in the retina with this exact same protocol that we're using today, antibiotic, you know, fix the terrain kind of thing in glaucoma.
Which is, you know, so there's some interesting things there. One is that some people have found in the retina Chlamydia pneumoniae.
Oh yeah, no question.
Okay. So the organism can show up there, but also amyloid will show up in the drusen in the back of the eye.
That's Don Anderson out of UC Santa Barbara, who published that work on the beta 142 and drusen. Yep.
Right. So it sort of fits together that would be also a clue with the eye of what's happening here.
Yeah, it's a screening tool, and I think people should take it. I mean, you know, there are more eye diseases out there than neurodegenerative diseases to some extent, and your eyes are just as important. What Dr. Tremp, and I'm treating myself for toxo right now, and what Dr. Tremp said, and then I'll let you go, but we always have Dr. Harshfield land the plane. But what Dr. Tremp said is, the things he told me were so brilliantly simple. He says, Tom, you know, the difference between an eye doctor and a brain doctor is that you have two eyes. So, for example, macular disease, if you're a good doctor, statistically goes to the fellow eye 22% in 1 year, 85% in 5 years. So if you're a good doctor and it doesn't spread to the second eye, you've done your job. So right now, I haven't done it yet. I'm going to start microcurrent treatment on my right eye. I'm doing iodine treatment on my right eye, and I'm doing methylene blue systemically. But I'm just going to, I'm going to work, and my son's an optometrist, so he does detailed OCT on me. So what I'm doing, I'm a clinical study of one, but I'm going to just go on my right eye with eye-centric treatments, microcurrent and then iodine. And the iodine treatment's nasty, but it's, it's painful, but I'm doing it.
Wow.
Anyway, with that, any last comments, Dr. Balin? It was amazing talk, and I think more people have to be aware that this is at the root of so many conditions. You mentioned diabetes, atherosclerosis. You know, Dr. Barodi, who developed the triple antibiotic, we interviewed him, and he said, not in this, forum, but he said all his family died of heart disease, and they had Chlamydia pneumoniae infection.
Yeah,
hard to treat.
I think that overall, if I could summarize a little bit, is that we need to pay attention to these really polymicrobial issues that are happening in us or around us, and that really can now have drastic effects in keeping us healthy or diseased. And I think that, you know, when COVID hit and we started seeing the sequelae from COVID, that a lot of people, it's opened their eyes to, oh my God, you have something that can actually do something systemically and in your brain and lead to neurocognitive deficit in a number of people. Yep. And this brain fog and long COVID, et cetera. And that's really giving us more of a global issue here of how infectious agents can work. And for us to not take this to the point where we need to start making a difference here through diagnosis, look at biomarkers for this, get it early, is insanity. We need to start putting this into the repertoire of a physician, so that, you know, if you're showing up with any of this, or along the way, as you go to your healthcare provider and you're relatively healthy, is that they should start testing. They should start routine testing along this this avenue as well.
That's a long conversation, Dr. Carter. I didn't, because you work so hard in the Alzheimer's space. Before Dr. Harshfield lands the plane, do you have any comments for Dr. Balin?
No, fabulous presentation, and of course, when I'm doing a lot of the vibrant infectious panels on people, we're finding a whole lot of pathogens out there, which, again, these things are harbored in the vast majority of us. I don't think I've ever come across anyone that doesn't have something there, right. And especially the IgG levels and oftentimes, of course, IgM and so forth. But it really kind of goes back to, the expression of that really goes back to, immune resilience. And that's what we really all have to work on.
Yes. The other thing that I wanted to mention is vaccinations are good. Vaccinations have been shown to be somewhat older people that have had multiple vaccinations for a variety of different issues, from Shingrix, for shingles, to pneumococcus, to the flu vaccines, et cetera. Actually, it's been shown that there's a decreased risk for developing dementias. So there's different theories behind that, why that's working that way. But I actually think that that's a really good thing. There's probably crossover protection or immune activation that is now working more globally in many of us.
Well, maybe we should be vaccinating our older folks and not the younger folks. And there might be rather than just, make it a money grab, if we can find out, the specificity of it and use it in a targeted way rather than blanket everybody, 95% who don't need it or whatever. I'm just being a little bit...
But that's true. There's some evidence with the IVIG approach that's been taken with Alzheimer's disease clinical trials. Mm-hmm. So there's the limited efficacy with some of that. However, if you do the IVIG approach, I think for infectious agents, you've got another story. You've got a way of actually protecting against some of these pathogens that we've identified already.
Right. And with that, Dr. Harshfield, land us.
Dr. Balin, thank you. I think maybe to kind of focus on what we've learned, because we always go down these very important little narrow streets to talk about specific things. We were talking about the microglia in the brain and inflammation and infection. And as an interventional radiologist, I did a lot of angiography and saw plaque formation in the brain. And we did a lot of venography. And we saw these jugular veins as they exited the skull were starting to narrow. And we misunderstood that as if the venous narrowing was like an atherosclerotic plaque in an artery. And we did balloon dilation and stenting and all that didn't work. Turns out both atherosclerosis and this venous stenosis that happens around the brain is because of a toxic extracellular fluid, as you commented upon. That's where the tau, the beta amyloid is excreted in the extracellular space. The tau hangs around in the intracellular space. They both are kind of the shotgun shells of the immune system. They're mad at something. And we know that these Alzheimer's issues and dementia are caused by toxins and trauma and infection and inflammation. And all those things can create this overreaction, too many fire trucks, as Dr. Lewis alluded to. We treated a bunch of COVID folks with inhalation of amnion tissues, nanoparticles, exosomes, those sorts of things. And it talked about the way that we get that, as you alluded to, through the neuroepithelium when you breathe it through your nose and into your lungs, and then it gets into the vascular system and goes across the blood-brain barrier. So we gave intravenous and inhalation of these amnion products, and these folks did great. And guess what? They started remembering their grandkids' names. So in addition to cardiovascular effects, we were improving their brain and mentation. Mm-hmm. What those amnion fluids and products do is that they educate the macrophages, one of the things they do, from the M1 macrophages, like in adults that only know how to scar, they turn them into M2 macrophages that know how to resolve inflammation, to heal in that fashion. So this is a beautiful description of how this works. This is an infection that can institute Alzheimer's and neurodegeneration. A lot of these folks that have traumatic brain injury, we have a project going on with the NFL with chronic traumatic encephalopathy. These boys have had a long-term bout with this. Their brains are small. We can see it on an MR. That's not good. We want to know this 30 years ago, to your point about biomarkers. And we're going to do CSF evaluation of about 3,000 biomarkers in the cerebrospinal fluid and these older guys. And we want to be able to extrapolate that back to what would have, if 30 years ago, right when they quit the NFL, if we could done a spinal tap, saw this and this and this, we could have predicted, hey, you're going to have that. So anyway, it's great. I appreciate the heck out of this. And again, it's kind of the pathogen, the cause of disease. Is it the pathogen or is it the terrain? Their pathogens are going to always be there. So let's concentrate on what Dr. Lewis teaches us about. Get your terrain healthy. Get that vascular bed. Keep that healthy. Keep that extracellular space and fluid as healthy as possible. And then when we have these things, to be thinking, okay, what's the pathogen causing this? And the treatment's not going to be an antibiotic. It'll be something probiotic. And also the thing that really scares me, the literature now is in your favor now, Dr. Balin, because for 20 years, they said that this idea that the beta, tau, and amyloid proteins were the cause of Alzheimer's. That was falsified data that came out 6 weeks ago. So for 20 years and billions of dollars, they've misdirected us. They are now understanding what you've been teaching for all this time. I mean, good for you. I know it hadn't been an easy path, but I want to just say I appreciate what you did. Thank you.
Thank you. Thank you. I certainly appreciate that as well.
And Dr. Carter, he says it the same way every time. It says, people just need to understand, and your traditional doctor doesn't have the analytics to tell you this. How full is your bucket? And that really dictates sort of how long you can stave these things off. You won't be able to stave them off forever, obviously, but you will be able to stave them off. We have one hand up, and I'm a sucker now for this. So we'll let Mary, you want to throw a comment in, and then we'll let Dr. Balin get back to his work. And you're so much gratitude for the info. Do you have a comment?
Your audio's off.
Let's see.
Click that mute button in the bottom left, Mary.
See, there's my IT guy.
I think I've done it. Is that it?
Yes, go ahead.
Yeah, I just want to thank you all for such valuable information. I need to get on this and start my testing and figure out what all's going on, because like you say, the earlier the better. So I just want to thank you all. And I truly appreciate all the information that you're giving to everybody.
Right. And if you're not testing, you're guessing. And that's the toughest thing,
Right.
you know.
Right.
So that's where we need to go. We need to convince people that it's not prevention. You know, our whole program that we've revamped since I met you in Switzerland, Dr. Balin, is where do you lie on the health disease continuum?
Right.
Okay. And in that concept, there's no such thing as prevention because the diagnosis is an artificial point. So it's all about moving yourself in the better direction. So you can throw out the term prevention and just say, look, I need to get health. Everyone on this planet has to get healthier. And with that,
very true.
Thank you so much. Love to have you back sometime and love to carry on a conversation with you because it was really rewarding. Appreciate your time.
Well, thank you for having me too. Good
really
to meet everybody on this.
Take care. Bye for now.
Okay.
Bye.
Bye.
Thank you.
Thank you for the good information. Thank you.